Hello, and welcome to the fifth talk in the Mathematical and Computational Ophthalmology seminar series. My name is Paul Roberts, and I'm a Centre Fellow in the Centre for Systems Modelling and Quantitative Biomedicine at the University of Birmingham, where I work in the mathematical modelling of the eye. I'm the creator and host of this seminar series, together with David Crabb, who heads the Crabb Lab at City University of London, and Phil Luthert, a Professor of Pathology at the UCL Institute of Ophthalmology. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical and computational communities, raising awareness of the exciting research being conducted in the field and encouraging future work in this area. Do subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. These are also listed on our worldwide page, the link to which can be found below in the About section. And our thanks go to Worldwide for instigating this platform for sharing scientific research. Before introducing our speaker, Ross Ethier, I'd like to invite participation in an Association for Research in Vision and Ophthalmology, or ARVO, Special Interest Group, or SIG, in Mathematical and Computational Ophthalmology, to be held via Zoom on Tuesday the 12th of September from 1 till 2:30 p.m. British Summer Time. This SIG, organised by David, Phil and myself, will consist of 3 brief talks, followed by a time of structured discussion. The aim of the SIG is to bring together quantitative scientists, biomedical scientists, and clinicians using quantitative methods in ophthalmology, to summarise the current state of affairs and to clarify future directions, fostering new collaborations. Please do register your attendance on the ARVO website, the link to which can be found below in the About section of this YouTube channel. I'd also like to remind our listeners that David Crabb, Phil Luthert and I are guest editing a special issue on mathematical and computational ophthalmology in the journal Investigative Ophthalmology and Visual Sciences, or IOVS. We welcome submissions of original research applying quantitative methods to examine any aspect of visual system structure or function. The deadline for submission is December the 1st of this year, 2023. All submissions will be peer reviewed and publication fees will apply to papers that are accepted. A link to the IOVS special issue advert can be found below in the About section of this YouTube channel. I coined the term mathematical ophthalmology to denote the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease, while computational ophthalmology is an existing term used to denote the field which uses computational algorithms, and in particular AI, to preserve vision by identifying and predicting ocular features and risks. Thus, the focus of mathematical ophthalmology is upon elucidating mechanisms, while that of computational ophthalmology is upon prediction and identification. In our last seminar, focusing on computational ophthalmology, Tiernan Keenan gave a fantastic talk on diverse applications of artificial intelligence and mathematical approaches in ophthalmology, the recording for which can be found below in our YouTube playlist, together with recordings of all previous talks. This week, we're switching back to mathematical ophthalmology with our speaker, Ross Ethier. If you'd like to ask Ross a question, then please do send these in via the YouTube chat, both during and after the talk. The talk will last for about 45 minutes, followed by 15 minutes for Q&A, in which I will relay your questions to Ross. So it gives me great pleasure to introduce Ross Ethier. Ross is the Lawrence L. Gellerstedt Jr. Chair of Bioengineering and Georgia Research Alliance Eminent Scholar at the Georgia Institute of Technology and Emory University. He has undergraduate and master's degrees in mathematics and engineering from Queen's University and the University of Waterloo in Canada, and a master's and PhD in biomechanical engineering from MIT. He's received numerous prizes and awards, including the H.R. Listener Medal for outstanding achievements in the field of bioengineering, a BioE Outstanding Advisor Award, and the Lewis Rudin Prize. He's a fellow of a number of societies, including the Association for Research in Vision and Ophthalmology and the Biomedical Engineering Society. Ross serves in numerous ways, including as an ARVO glaucoma section trustee, and is on the BMES Board of Directors and the ASME Committee on Government Relations. He's an editor for 7 journals and is currently co-editor-in-chief of the Journal of Biomechanical Engineering. His research seeks to understand the biomechanics and mechanobiology of ocular tissues, with a particular focus on the pathogenesis of glaucoma, as well as the biomechanics of the sclera and its role in myopia. So I'll hand over now to Ross to present his talk entitled Computational and Mathematical Approaches to Myopigenesis. Ross.
Well, thank you so much for your kind introduction, and congratulations to you, David and Phil, for establishing this seminar series. I think it's a great initiative, and I'm delighted to be able to participate in it. And I hope that you're now able to see my first slide. Could you please confirm that you can hear me and see that slide okay?
Yes, that's all good. Thanks, Ross.
Perfect. Okay. So as Paul mentioned, we're going to talk today about myopigenesis, and I'm cognizant that the audience has a diverse set of backgrounds. So some people may know a lot about myopia and others may know less. So I'll sort of bring you along through the physiology and what we know about that and the biology, and then talk a little bit about the math side of it. So let me just advance a slide. I have no relevant disclosures or conflict of interest in this talk, and I want to begin by acknowledging and thanking my collaborators and trainees on this project. Most of the work that you will see today arose from the PhD thesis of Dylan Brown, a very talented student who was co-supervised by myself and my longtime colleague, Michelle Pardue, who really is the card-carrying expert on myopigenesis. And as you can see, there's others involved in this project. I'd just like to highlight Maureen Kane, whose work will figure later on in the talk. Saptarshi Chatterjee, a postdoc in my lab. Rodolfo Repetto, a great collaborator in Italy who I enjoy interacting with. And Maria Dvorshna, who is just moved to Edinburgh and has helped out with some of the modeling that's been done. So let's begin with a little bit of background on myopia. So this is the most common refractive error. And in fact, by 2050, it's predicted that half of the world's population will have myopia. And it's quite interesting. If you look at this picture here, this is from a Nature review article, you can see that every individual who you can see clearly is wearing glasses. And in fact, in some countries and some age groups, it's the incidence of myopia is something like 95% or more. So what happens in this condition? Stated simply, it's the eye's optical properties are mismatched with the axial length of the eye. So in normal or clear vision, the incoming light is focused and that focal point is coincident with the location of the retina, and you see a sharp image. In the myopic eye, the focal plane of the incoming light actually lies in front of the retina. And so what is perceived by the retina is blurred. And in the vast majority of clinical cases, this situation is due to excessive axial eye size. So the eye is simply too long. And as you could well imagine, to reach that situation of being too long, there has to be abnormal growth of the sclera, the white outer coat of the eye. And so there's a lot of remodeling that happens in the sclera, and we'll talk quite a bit about that as we go along. So why do we care about myopia? As you know, there are treatments for it, spectacles, etc. However, what some people are less aware is that even though you can optically correct the vision in a myopic eye, there are long-term risks associated with myopia, particularly high myopia. And in particular, myopia is a risk factor for glaucoma and other potentially blinding diseases. And so when you put that fact together with the incoming incidence of ever higher rates of myopia, you're actually looking at a bit of a public health tsunami coming down the pipeline as associated with ophthalmic conditions. And so we really do need to better understand the process of myopia development or myopigenesis. So to better understand that, I want to mention something called emmetropia, which you can think of as the condition of I don't need glasses. And suppose that you had a situation in which the light was focused behind the retina. So you're going to obviously have a blurred visual stimulus. We know that triggers retinal signaling, that retinal signaling in turn induces remodeling of the sclera and causes the eye to elongate anterior and axially. And so what happens is that as the eye grows, eventually the focal plane of the light and the location of the retina come into register. And then at that point, the eye will actually stop elongating. And so it's clear that there's some sort of a feedback loop. And we call that process emmetropization. And basically in emmetropia, the axial length of the eye matches the eye's optical properties. So you can think in some ways of myopia being a failure of the emmetropization process. And I like to show this graph. It's quite interesting. This is from a large longitudinal study of school-aged children, and they measured the axial length of their eye each year for multiple years, and also their refractive status. And at a certain point, they were able to classify the children as either being emmetropic or myopic. And you can see, if you look at the emmetropes, these children's eyes are growing over time, and that's as expected because the child is developing and growing. But you'll also notice that the myopes are growing at a much faster rate, or an appreciably faster rate. And what's really interesting is that time zero on this graph here is actually the year at which the myopes were diagnosed as having myopia, based on their refractive error. And so you can see that even before a clinical diagnosis of myopia, you have an elongation of the eye. And so there's some sort of a sustained drive for the eye to grow longer in these myopes. Now, why does this happen? Well, the short answer is we don't really know, but we do have some information about that. And probably the best accepted theory is that this occurs in children and adolescents who spend insufficient time outdoors. And that's kind of a complex story, but I think probably the most widely accepted part of that story is that ambient light levels are very important here. In other words, even in a well-lit room inside, the light levels are much less than what you would experience outside, and exterior light levels seem to be anti-myopigenic. There are also other things that occur outside versus inside. The spectral content of the light outside is different as compared to interior light. The spectral content of the scenes that you see is actually different, and you tend to do more near work inside, so that you're reading or doing something quite close to you, and so that implies a sort of a sustained accommodation, and there's also some peripheral defocus that happens with that. The gray shaded text here refers to reasons that are maybe less well accepted or understood. There is a genetic component, and then there's also some data that suggests that inflammation and oxidative stress plays a role. But really, I think at the end of the day, most people would say that it's a bit mysterious. And so we really need to know more about the process of myopigenesis. Because it's such a complex process, we rely significantly on animal models of myopia to understand that. And these have been used for many years. You can see there's a number of species that have been used. Today, we're going to mostly talk about mice. And how would one induce myopia in a mouse? Well, what you want to do is you want to defocus the image on the retina. And so one way to do that is to put goggles on the mouse, and in the goggle you place a lens or a diffuser which blurs the image on the retina, and this is mounted onto a head pedestal which is attached to the skull. And those goggles can either, as I mentioned, incorporate a diffuser, and when we do that, this is called a form deprivation type of myopia. The image is just quite blurred. Or you can actually use a negative lens that pushes the focal plane of the image behind the retina, and that's called a lens-induced myopia. And here's what it actually looks like in a mouse. So I am going to talk quite a bit about the sclera today, the white outer coat of the eye. And this is a largely avascular connective tissue. And as you might well expect, the major constituents of that tissue are collagen, which gives it a lot of structural integrity. And then there's also a lot of proteoglycans and glycosaminoglycans that we know also play important metabolic and functional roles in terms of the biomechanical properties of the tissue. And then there's a population of scleral fibroblasts also within that tissue. So let's look a little bit at what happens to the sclera when myopia is induced in an animal model. And this is work from the Norton lab at UAB in tree shrews. This is a small animal that's actually quite closely related to primates. So we think this is a good model of what's happening in human. And this is a rather complex graph, so let me try to walk you through this. In the horizontal axis, you have days of visual experience. So basically, day 0 is when the animal opens its eye. So this is how long the animal has been seeing for. And at day 25, they put a diffuser over one of the eyes. These experiments are almost always done unilaterally, so the contralateral eye serves as a control. And they've got 2 sets of data plotted on this graph. So let's break that down. On the right-hand axis, they plot the axial elongation rate of the eye. So they measured the length of the eye, and this is the percentage it's elongating per day. And what we're seeing here are measurements from a group of naive animals. So nothing was done to these animals. And basically what you observe is when the animal is very young, its eye is elongating at quite a fast rate. And then as it ages, it sort of asymptotes towards a steady state where it's not really elongating very much at all, but still growing a little bit. If we look at the contralateral untreated eyes, so the control eyes, you can see there's something interesting that happens here, but these data points are relatively close to those of a naive eye. But look at what happens in the eye that was treated by placing the diffuser over it. You can immediately appreciate that the elongation rate is much higher than you see in the contralateral control eyes and stays higher for the duration of the measurements. They also made biomechanical measurements on the sclera. So they're looking here at the scleral creep rate. So you dissect small pieces of sclera and apply a constant force to it or stress to it, and then measure how it slowly elongates. And that's characteristic of a viscoelastic response of the tissue. And again, here are the naive animals. And you can see that the control animals are quite close to that naive cohort. But notice in the eye with the diffuser on it that there is a much higher creep rate, and that's indicative of remodeling of the extracellular matrix of the sclera in these treated eyes. And that is consistent. They also did a similar set of experiments with negative lenses, and the general story is the same there. Now, what could be mediating this remodeling process within the sclera? Well, they measured the levels of various MMP, actually message, excuse me, for MMP. So they looked at MMP1, and you can see here, so the dashed line are sort of control or normal conditions, and the filled symbols indicate the levels of message that they were able to assay in the treated eye versus the open symbols in the control eye. And I think you can see that there's quite a significant jump in the levels of this particular MMP. The MMPs, if you're not aware, are enzymes responsible for remodeling, breaking down the extracellular matrix. They actually looked at several MMPs and saw changes, and they also looked at the TIMPs, which are regulators of MMP activity, and in some cases saw changes. So it's quite clear that there's quite a lot of enzyme activity going on within the sclera of these eyes as they undergo myopigenesis. And in fact, there's even been postulations that alterations in scleral fibroblast mechanobiology may underlie the excessive axial lengthening that one sees in myopigenesis. So the takeaway from this is that the sclera is really a key player in this entire process. In fact, there's now a number of studies, some of which are summarized here. Basically, you have loss or increased degradation and downregulation of proteoglycans and glycosaminoglycans within the sclera, and changes in the crimping and amounts of collagen within the sclera. Okay, so it's a complicated story, as many things are, and so we want to understand it better. And for this purpose, we're going to use mice, which are not as widely used as some of the animal models, but we like the mouse model. There's a number of benefits to using the mouse, probably the most salient of which is you have very precise genomic control over these animals. You can knock in or overexpress genes that you think might be important in myopigenesis. But using the mouse comes at some cost. And one of the costs is there are some anatomical differences. So for example, the mouse eye has a lens which, relatively speaking, is much larger than in the human eye. But probably the biggest problem with using the mouse eye is it's just so darn small. And the sclera presents a relatively small amount of area, as you can imagine, and also it's quite thin with an average thickness of only 70 microns. And that means that if we want to do tests on the sclera, we have an experimental challenge. If we want to do a biomechanical test, for example, it's just really hard to work with a piece of tissue that's so tiny. And so that motivates some of the work that I'm going to present to you today. We first wanted to develop a methodology to actually quantify the biomechanics of the sclera in a mouse eye. And you'll see that we use something called an unconfined compression test, complemented by some nice modeling work. Then once we have that test going, we want to use it to measure scleral biomechanical changes that are associated with myopia. And for that, we'll use form deprivation. And then we're going to talk about a molecule called all-trans retinoic acid, or atRA, which turns out to be quite interesting in myopia. And finally, I'll just very briefly describe a little bit about some modeling we've done of atRA transport in the eye. So let's start at the beginning. We'll talk about the methodology, and we're here using something called a cell scale micro squisher device, which is shown here. And the business end of that is this temperature controlled bath in which you place your sample to be tested. So here's a zoomed up view of that region. And you have your sample is shown here. So this would be a small piece of sclera shown in blue, and it's sandwiched between two rigid and permeable platens, the upper of which is attached to a cantilever that can be actuated up and down by this piezo motor here. And so what we do is we squeeze that sample. This cantilever moves downwards and squishes the sample. And in so doing, the sample expands laterally, and there's nothing to stop it from doing, hence the name an unconfined compression test. And the whole while this is being visualized by this camera here. So this is a movie, which I hope you can see, that shows this. This is actually not a piece of sclera, but it does demonstrate the principle nicely. So we have a measurement of axial strain or deformation in this tissue or this sample here. And also this cantilever is calibrated so that we know the force that is being applied. It's kind of like AFM, if you're familiar with that. So how do we actually do the experiments? We're using freshly enucleated eyes from wild type black 6 mice. We keep the eyes submerged in PBS throughout the process. And basically we open the eye, and specifically the sclera, by creating this butterfly shape here after the eye has been enucleated. And then using a 1 millimeter diameter tree fine, we punch out 4 scleral samples as shown by the red circles. And we coat the top and bottom of those samples with graphite powder. That is because when we do these unconfined compression tests, we want to allow the sample to expand freely in the lateral direction. In other words, orthogonal to the plane, the direction of compression. And so we want to minimize the friction between the sample and the platens, and graphite powder is a great way to do that. So here's schematically, this is what one of our little samples looks like. It's kind of like a little hockey puck. And we do a preconditioning protocol that I won't get into, and then we do something called an unconfined stress relaxation protocol. So here's our preconditioning. This is the applied force versus time, but this is the important part. This is the compressive strain, which is applied to the tissue. And you can see that we have 3 steps. In the first step, we compress the tissue by 5%. Then 10%, then 15%. And we monitor that tissue as it reaches an equilibrium state at each of those 3 steps. So now I'm going to show you a brief movie. This is a numerical simulation just for visualization purposes, and we're seeing a small sector of kind of a approximately cylindrically shaped scleral sample. And you can see it's been cut in a funny way just to allow you to see inside the sclera. So the force is going to be applied in this direction. The sclera, you will see, will elongate as it moves outwards in this direction, as shown by my mouse pointer. The contour lines, colors will show you the fluid pressure within the sample. The arrows will show you the direction of fluid flow. And just to say that the tissue is poroelastic, so you can think of it as a solid matrix in which the pores are filled with water. And as you squeeze down the tissue, of course, the water is going to be expelled out of the tissue, as you see. So let me just start the movie now. So this is close to the beginning, and you can see that initially there's a very high pressure within the scleral sample. And then as water is exuded from the edges of that sample, the free edges of that sample, the pressure declines over time. And of course, that pressure is supporting the force that's being applied. So I'm just going to play that movie once again. You can see, if you look down here, that the sample expands laterally quite a bit and then sort of relaxes back. And so the force versus time that is applied to the sample to keep it at that constant 5% strain is actually time varying. So let me show you actual data now. So here's what we seek to achieve in terms of the loading of the sample. Here's what actually happens in real life. So we have 3 loading steps, a 5, 10, and a 15% axial compressive strain step. And you'll see that it's quite close to the graph on the right, but not quite the same. There's a little bit of an overshoot, and there's also quite a bit of jitter. And this has to do with some limitations of the testing system that I won't get into. But I would ask you to remember that jitter because it becomes important later on. So we impose these 3 levels of compressive strain, and then we measure the force from which we calculate the applied stress on the sample. And you can see, as you would predict intuitively, when you first compress the sample, the stress jumps up quite significantly and then decays over time. And so it's this time or stress versus time data set that we're going to analyze to extract mechanical information about the scleral samples. Now, how do we do that? So we're relying completely on this beautiful paper from Soltz and Ateshian. And let me just walk you through the key elements of that. They developed or used something they called a biphasic cone-wise linear elastic model. So here's our tissue sample again between 2 platens. It's being compressed and fluid is coming out the side. So the key element here is that we assume that the tissue sample has a bimodular response. So if I were to plot the stress strain behavior of that tissue in a simple uniaxial test, I would see a different effect of Young's modulus in tension, as shown in blue, as compared to what I would see in compression, as shown in red. And this is pretty typical of tissues. And I'll just give you advance notice that the parameters that we will seek to extract from this tissue through the modeling are a compressive Young's modulus, which we denote by E minus Y. And there's an interesting thing about the sclera, which is that the collagen fibers lie pretty much in plane. So if you think about how this test occurs, the resistance to compression is not really going to depend on the collagen. Instead, it's going to more depend on the ground substance around the collagen, which is primarily proteoglycans and glycosaminoglycans. So getting this E minus Y, this compressive modulus, really tells us about these proteoglycans and glycosaminoglycans. On the other hand, in plane, the loading is tensile. And so we will also extract a tensile modulus, and that really tells us about the collagen since it lies in plane. We also have fluid transport within the tissue, and so the hydraulic permeability of the tissue turns out to be an important parameter. And then there's a 4th parameter that comes out of the modeling, as you'll see, and that's a little bit harder to physically interpret, but it's basically showing coupling between radial and axial effects. So if you're interested, this is documented in this paper here that was published several years ago. And I'll just go through the basics of the theory at a fairly high level. And this is very closely following the Soltz and Ateshian development. So we're using poroelastic theory. As I mentioned, the tissue is modeled as a solid matrix, which supports a stress SE for the elastic stress. And then the pores of that matrix are filled with a fluid, which is assumed to be incompressible. And so there's also an isotropic stress due to the pressure from that fluid when you add those together, you get a total stress. We model flow within the tissue using Darcy's law. K is the permeability tensor, W is the relative velocity of the fluid with respect to the solid matrix. And then we satisfy force balance in the absence of body forces and inertial effects. That simply takes the form that the divergence of the total stress tensor is 0. And we also impose a conservation of mass on the fluid, which can be written in this way. So this is all pretty standard. The interesting part is the formulation of the strain energy density function for the solid matrix, which gives us this S superscript E. And basically, this comes from quite a complex paper from Kernier and colleagues. But what they do is, if you're not familiar with this, this capital A is the so-called structure tensor. These E hat vectors point in the direction, the preferred directions within the solid matrix. And by taking this product, this tensor contraction product between the structure tensor and the strain tensor E, you extract a scalar value which is equal to the deformation in the direction of this EA vector. And of course, the subscript A can be 1, 2, or 3, representing the three directions. And what Kernier et al. Did was they said that if that tensor product was less than 0, then the lambda would take a value lambda minus, and if it was greater than 0, it would take a value lambda plus. And so this reflects the behavior, the differing behaviors of the material in tension versus compression here. And this formulation, if you're familiar with solid mechanics, is a little bit like the Lamé formulation. It's actually a generalization of the Lamé formulation, where the lambdas and the mu's are the Lamé constants. So for the type of loading that we're imposing on the material, the green Lagrange strain tensor looks like this. This is pretty standard theory just from the kinematics of the deformation field that we expect for a thin sample. And then the problem is that you actually have 12 coefficients in that constitutive model for the solid matrix, which is way too many to deal with. And so what Soltz and Ateshian did was they assumed a form of isotropy in these different coefficients. So they have a single coefficient that describes the behavior of the tissue in compression, a different coefficient that describes in tension. This is the off diagonal, and then this is the kind of like a shear modulus. And at various points, it turns out that it's convenient to rewrite these 4 coefficients into some linear combination. It just makes the mass a little bit easier. So you'll see things like H minus and H plus, or E minus and E plus. Those are just moduli in compression and tension, respectively. And it's justified to do this because of this. Effectively, you have a separation of the behavior of the tissue due to the fact that the collagen is in plane with the scleral sample and it's under tension in that direction, and it's not in plane in the direction that's being loaded. And so Soltz and Ateshian tested this in cartilage, and it worked very well. So we thought it would be good to use this also for the sclera. So this is the force balance equation. So here's the terms arising from the stresses within the solid matrix. And then there's two terms arising from the Darcy contribution. And they represent the motion of the solid matrix and the fluid, respectively. And then basically, if you assume small displacements and strains, it's convenient to work in the Laplace domain. And the bottom line is that you can analytically solve the governing equations. And the net result of that is a transfer function, which you can interpret as a modulus of the tissue. So it's the ratio of a Laplace transform of the stress over a Laplace transform of the strain. And this is the axial stress. So it's the total force applied to this sample in the axial direction or in the vertical direction, divided by the initial cross-sectional area of the sample. The over bars actually denote normalization with respect to the equilibrium values. That just makes the math a little bit easier. And your Laplace variables are down here. And the important thing is that this analytical solution depends on 3 constants, A, B, and C. And those constants have incorporated within them the material properties of the solid matrix and also the permeability of that solid matrix. And so essentially what we do is we have experimental data for force and displacement or axial strain. So we experimentally can determine G bar. We have this analytical solution. We attempt to fit the analytical solution to the experimental data. And the free parameters in that fitting are these quantities here, the H plus, H minus, etc. So it's quite a messy nonlinear fitting process, but you can extract material parameters from that. So let me show you how that works in practice. Here's the normalized axial strain and the corresponding normalized axial stress imposed on that sample. So we Laplace transform these quantities. We get a transfer function. And here I'm showing the results of the fitting. So the green dots here are the experimental data. So we're plotting that transfer function, both its phase and its gain here. And the red dashed line shows the fitted model. And I think you would agree that the quality of the fit is really quite good. Now, of course, there's always a but on these things. And I purposely didn't show you all of the data before revealing it just now. And you can see that there's actually quite a bit of noise. And I would ask you to recall that jitter, which I had previously mentioned. This is a high frequency jitter, and this comes about due to basically, it's the way the control system works for the testing device. So when we do the fitting, we only do it up to a certain frequency. And we kind of throw away the data above that because it's just corrupted by this experimental artifact. So when we do that fitting, one measure of the quality of the fit is looking at an R-squared value for the fit, and you can see that each dot is one scleral sample, and it's quite good here. Average value is 0.99 something. So when we extract those parameters. Here I'm showing this is the experimentally measured normal strain on the tissue sample, and here we have a comparison between the experimental data and the fit, and you can see I think that the fit is quite good. So we're very pleased with that. So this process seems to work very well, and we were happy with it. But the question then arises: does it give us results which are comparable with what one would obtain from different approaches? And unfortunately, it's not possible to make measurements, or we haven't been able to do it, and others haven't done it in the mouse sclera. So instead, what we did is repeated the same experiments with pig sclera. And so here's again an example of the fitting. And what we're plotting here on the right-hand side, this is the in-plane tensile modulus of the scleral sample. The open symbols here are for pig sclera, and the shaded are for the mouse. And you can see there's apparently a systematic difference between pig and mouse sclera, at least as far as its tensile modulus goes. And we also have the hydraulic conductivity measurements. And these values here refer to the axial strains. Remember, there's a 5, a 10, and a 15% strain step, and we extract data at each of those steps. And as you would expect, the modulus increases as the strain goes up. So we see, nonlinear stiffening behavior, and the permeability decreases as the tissue sample is compacted. Now, luckily, there are measurements in the literature for pig sclera, and this is the results from a biaxial tensile test. The mean is the line, and the shaded region is the confidence interval around that. And the confidence interval is broad, but it agrees quite well with our measurements, so we feel that's good. And there's also measurements of the permeability. The agreement is not quite as good here, but this is a notoriously hard measurement to make. So we thought that the agreement was acceptable here. We've done some retest variability measurements, et cetera. And overall, we believe after developing this approach that this is giving us a good characterization of the mechanical properties of the mouse sclera. So then the question arises, what would one do with that? And so what I want to do now, and I'm cognizant that there's only about 10 minutes left, I'm going to describe to you how we use that approach to measure changes in the mechanical properties of the sclera during myopia. So this is the experimental design. We're using C57 Black 6 mice. Again, we fit them with goggles with a diffuser lens unilaterally. And we follow those animals longitudinally for either 1 or 3 weeks of form deprivation, during which time we make measurements, OCT to measure ocular dimensions. We measure the curvature of the cornea with a keratometer. We also measure the refractive status of the eye with an autophoto refractor. At the end of the experiment, the animals are sacrificed and we do biomechanical testing on the eyes. So we actually have several cohorts. One is with the unilateral form deprivation, and then we have naive control animals, which are untouched. So the first question is, do we get a myopic shift in these animals in our hands? And the answer is yes. This is the refractive shift compared to baseline here. This is the baseline data. These are the naive animals, so there's not a net myopic shift between the two eyes, but you can see at 1 and 3 weeks of form deprivation, we have a clear myopic shift of about 4 diopters. So yes, we are inducing myopia in these animals. What happens to the sclera? Here we're showing the tensile stiffness of the sclera. The naive animals are in gray. The control animals, are the control eyes, excuse me. This is the contralateral eye, so the ungoggled eye is in blue. And then our 1 and 3 weeks of form deprivation are shown in the 2 colors of orange. And I think you can see the 5, 10, and 15% strain levels, particularly at the higher strains, there's a clear loss of tensile stiffness in the myopic eye. So the sclera is simply becoming softer in plane. And conversely, there's an increase in the hydraulic conductivity or permeability of the sclera. And that's entirely consistent with what we expect. It's a little more striking if you look at the differences compared to baseline. So here's the control animals or the naive animals. You get about a 35% reduction in tensile stiffness, 3 weeks of form deprivation myopia, and about a 200 and some odd % increase in permeability or hydraulic conductivity. And that's quite a lot. And this is consistent with what we expect, right? We know that there should be turnover and loss of collagen, and also extensive remodeling of the glycosaminoglycans and some loss of those as the eye is elongating. And in fact, we were able to measure loss of some proteoglycans. This is dermatan sulfate, although interestingly, chondroitin 4 sulfate levels were not affected. So we have a nice story here where we're able to actually measure these biomechanical changes in the sclera. And it seems to be a good readout of what's going on in terms of scleral remodeling. Now, I'm going to jump to talk to you about all-trans retinoic acid, or atRA. And you might wonder, what is atRA and why is he interested in studying it? Well, atRA it's a vitamin A derivative. It's one of the retinoids. These molecules are very important in the visual cycle and also in tissue morphogenesis during development. But our particular interest is that in some studies in the past, and mostly in the chick, but also in the guinea pig, atRA has been implicated as a possible player in the development of myopia. And here the idea is, we know that a defocus on the retina will lead to scleral remodeling, but the question is, how does that signal get to the sclera? Remember that in between the retina and the sclera, you have the retinal pigment epithelium and the choroid, not exactly easy tissues for a signaling molecule to get across. And so the idea here is that, schematically, here's the retina, here's the sclera, we have these intervening tissues, we have a myopigenic visual cue. There's some sort of a retinal scleral signaling cascade, which ultimately leads to scleral remodeling. But we know very little about this. And we ask the question, what molecules are carrying this information? And as I mentioned, it's thought that atRA might be one such molecule. So we did kind of a simple, naive experiment, I would say. We're using mice again. And at P28, 4 weeks of age, we feed these animals atRA. So more specifically, we put atRA in a sugar pellet, or control animals get sugar only. They're very happy to eat that material. And we run them out for 1 or 2 weeks, making measurements as before. We sacrifice the animals and do biomechanical testing. Now, the first thing we did was ask the question, when we feed the animals atRA, does it get into the eye? And I'll show you data on that. And then the second question is, does it induce myopia? So the first question, does it get into the eye? So at the end of the experiment, we collected tissue samples. We sent them to our collaborator, Maureen Kane, who's an expert at measuring retinoids. This is mass spec data. The black are from control animals, which were not fed atRA, and the red are animals that were fed atRA. This is the atRA peak here, and you can see it's increased, and particularly so if I point out that this red scale is on a much higher magnitude than the black scale. So we're definitely getting atRA to the retina and also into the RPE, the choroid, and the sclera, as you can similarly see. So it gets into the eye. And the question then arises, does it induce myopia in mice? And the answer is most definitively yes. If you compare the control animals to the atRA-fed animals. In fact, in 2 weeks, we get about 6 diopters of myopia, which is a pretty significant amount of myopia over such a short time, and actually more than we got with the form deprivation. Associated with that myopic status, we see a change in axial length, which if you plot it compared to baseline is statistically significant. And the really interesting thing to me is that the sclera is also changed. So we can see that there's a diminution in in-plane scleral modulus, indicative of the collagen component, and an increase in permeability. So compare the blue and the red symbols at the different compressive strain levels. This is indicative of a loss of glycosaminoglycans and proteoglycans, just as we saw in form deprivation myopia. In fact, if we compare the form deprivation changes to the sclera head-to-head against the atRA-induced changes, it's quite remarkable. So blue is control, orange is form deprivation, and then deep blue is atRA control, and red is atRA-fed. And you can see if you compare the red to the orange at every strain level, both for tensile modulus and permeability, the atRA-fed animals, in terms of their scleral properties at least, are indistinguishable from the form deprivation myopia animals. So in other words, amazingly, we are replicating form deprivation myopia, which is kind of maybe the gold standard way of inducing myopia, just by feeding these animals atRA. And so to me, this indicates that all-trans retinoic acid is an important signaling molecule in myopigenesis. And so just at the very end now, just showing you one more slide of results, we got very interested in modeling the transport of all-trans retinoic acid. And this is evolving work, and I'm not going to really get into the nuts and bolts of it, but I'll just say that we're in the process of creating a multi-species model of mass transport. And very simply put, we look at atRA, and we know that atRA comes out of the blood in the choroid, as shown schematically here. We look at the movement of atRA in the extracellular space from the choroid into the sclera. One of the tricky things about atRA is that it's highly hydrophobic, so it has an obligate binding partner. Here, the atRA is shown as the yellow star, and that binding partner is shown in blue, and it acts on scleral fibroblasts within the sclera, as shown in the right-hand side. So when we model the mass transport, we obviously are interested in atRA, its binding partners, of which one is high-density lipoprotein, also receptors on scleral fibroblasts, and we account for water flux within this tissue. So we have diffusion, convection, and then a number of chemical reactions associated with the binding or dissociation of atRA with its transport partners and its receptors. And so this takes the form of a series of coupled ordinary differential equations in space with source terms related to leakage out of the vasculature and actually endogenous generation of atRA by resident cells and uptake of atRA. So as I mentioned, this is ongoing work, but it's actually teaching us some stuff about atRA transport that I hope to share with you in future. So in summary, I hope that I have convinced you that compression testing and biphasic theory can be used to quantify scleral biomechanics in mice. Form deprivation alters the refractive error and, most importantly, the scleral biomechanical properties of the mouse eye. And that depends on how long the form deprivation is imposed for. And what we observe is consistent with what we would expect, which is reassuring. Very excitingly, all-trans retinoic acid seems to be centrally involved in myopigenic signaling. And so we think this mouse model is really a powerful tool for the investigation of molecular mechanisms underlying scleral remodeling and myopia. And we're working very hard to uncover this mysterious process. So I'd like to acknowledge, again, all of my collaborators and financial support. Thank you very much for listening to me today. And I'm very happy to try to answer your questions. And I will now stop sharing.
Well, thanks ever so much, Ross, for a really clear.
You may be muted, Paul. Oh, sorry. Oh. No, it's my end. Sorry.
Cool. Well, thanks for a really clear and fascinating talk there, Ross. So we've got some questions have come in from the audience. If you have any further questions, then do continue to type them in, and I'll ask them to Ross if we have time. So the first question here is from John Doe. And so they ask, are there differences in scleral creep rate between individuals with high myopia and those with low or no myopia? And if so, how might these differences be related to the severity and progression of myopia?
Yeah, so that's a great question. And I will interpret that question to mean that the questioner is asking about in human subjects, because we do know that there are differences in animal models. So in human subjects, unfortunately, I think that question is probably unanswerable. We don't have the ability to measure scleral creep rate in vivo. The other thing is that, myopia diagnosis tends to be a bit of a retrospective thing. So by the time the person is identified as being myopic, there's already been a lot of scleral remodeling. So I don't think we know the answer to that question, and I apologize if I misinterpreted it. If you meant in animal models, then there would be a different answer, which is yes, there is a difference. In terms of severity and progression, yeah, that's a great question. It seems to, there seems to be a burst when you first deliver a myopigenic stimulus. So, and you might have recalled that from the tree shoot data. There's, shortly after you put the lens on, the sclera avidly starts to remodel itself, and then that tends to sort of equilibrate over time. And so there is definitely a progression there. In terms of the severity, yeah, I don't think we know the answer to that.
Thanks, Ross. And then a further question from John is, as scleral creep rate is influenced by mechanical loading, what role might intraocular pressure fluctuations play in myopia development, and how can we assess their impact on the sclera?
Yeah, that's a great question. So you might be aware, another area of interest of mine is glaucoma, and so I'm very interested in intraocular pressure, and I think it does play a role. Their IOP fluctuations will load the sclera in a time-varying manner, obviously, and the thing is, I am not aware of an association between myopic status and increased IOP or increased IOP fluctuations. And so from the clinical point of view, I'm not sure that there's necessarily an epidemiologic association there. But just from a physical intuition perspective, yes, there must be an association, I would say.
And then John goes on to ask, are there any sex-specific differences in scleral creep rate and myopia prevalence? If so, how might these differences contribute to the varying rates of myopia observed between males and females?
Yeah, good question. I'm not aware of any data that speaks to that question. So I'm sorry, I can't answer that question, actually.
No worries. So there's a question from R.S. Chong, I think, who says, really nice visualizations. They're referring here, I think, to your 3D simulation you showed, the time-dependent simulation. And they asked, are the scleral tissues fixed? And how does fixation affect tissue hydration?
Yeah, good question. So the scleral tissues are not fixed. If they were, of course, that would drastically alter the mechanical properties. And so we specifically do all of the experiments on unfixed tissues. And that's why it's incredibly important to, we literally sacrifice the animals and nucleate the eyes, sort of deliver them post haste to the tissue testing apparatus and, start the tests.
And then John's asked, so scleral stiffness is known to be altered in myopia. How could variations in the BCLE model parameters potentially represent different stages of myopia progression?
Yeah, that's a good one. John Doe has good insight. Yeah, I guess so. Most of the testing, sort of the conventional testing of the sclera, comes from, there's a lot of strip testing that's done, right? So if you're not familiar with that, it's, a small strip of material is excised and then unilaterally stretched. Although there's also a little bit of inflation testing, if I'm remembering correctly. So that would be reflected in that E plus Y parameter. So it's the tensile effect of Young's modulus of the sclera. I'm not sure that others have fitted their data to that formulation. I will say that the formulation that we're utilizing in that cone-wise linear elastic formulation, it does assume linear behavior. Obviously, there's a discontinuity at the zero strain point. But in tension, it's linear, and in compression, it's linear. And of course, we're aware that the tissues are nonlinear. There's two kinds of viscoelasticity. The questioner may know there's a viscoelasticity arising from fluid transport within the tissue, but then there can be intrinsic viscoelasticity of the solid matrix, which we also don't account for. So, there are other ways of testing and fitting the data. And they each have their pros and cons. It's just that when one wants to use the mouse, because of the physical size of the sample, you're really kind of restricted in what you do in terms of a testing approach. And so that's why we went with this formulation.
Thanks. And then there's another one from John. He said, what are the practical implications of any observed relationship between all trans retinoic acid and myopigenesis for clinical practice, public health, or future research directions?
Yeah, so that is a really good question. So the retinoids are very tightly regulated. They're important signaling molecules, particularly in the eye. And you have to remember that myopigenesis occurs in children and adolescents, so they're actually still developing. So I think that it would be probably a bad idea to drastically modulate retinoid levels in the eye of a child. But I think that what we've got here is kind of the end of a ball of string that we can pull on, because those retinoic acid, the atRA is signaling something downstream. It's pretty clear to me, and it's acting on scleral fibroblasts. So we are very actively engaged in trying to understand what that signaling cascade looks like. And of course, the mouse is great for doing that. So we're R01 funded at the present time to try to answer this question. At the present time, it's way premature to think about clinical practice, but in an ideal world, we would be able to figure out some aspect that's scleral specific in the signaling cascade that could be, modified by delivery of an eye drop, for example.
So we've got another question here from VJ. So he said, sorry if I missed this. He says, does atRA influence other ocular tissues, for example, the cornea? And how would we isolate the differences between effects on sclera versus cornea on myopigenesis?
Yeah. Hey, VJ, thanks for coming on. I don't know the answer to that, actually. So we haven't made measurements on the cornea. All of our measurements have been on sclera. It's definitely in lots of places in the eye. In the feeding paradigm, it's delivered through the blood, obviously. There are I was just looking at a report the other day. There are reports, at least one report of a change in corneal properties during myopigenesis, which is quite interesting because, maybe that makes sense if you think about there being some signaling molecule or molecules they're being released by the retina. It's conceivable that those could actually be transported anteriorly and ultimately affect the cornea. So I don't think we know the answer to that, but I will say that at least in clinical myopia and also in the mice, most of the effects we see in terms of the change in ocular dimensions are in the sclera. There are cases of myopia that are associated with changes in corneal curvature, et cetera. But like the biggest change we saw in the mice was an increase in vitreous chamber depth. So, and that's mediated primarily by scleral growth. So I think it's mostly happening in the sclera.
And I have one last question. This is from John again. So he says, during your research, have you identified any potential novel targets for intervention or treatment strategies for myopia based on the computational models?
The answer is no, not yet, but we're working very hard on it. That actually comes back to the clinical practice public health question. I will disclose that this is work being done with Saptarshi Chatterjee, the postdoc in my lab, and Masha Maria, who's in Edinburgh now, actually, formerly at Oxford, and also Rodolfo Repetto in Italy. The model is still in a state of development. That's why I didn't show any results from it. We are getting some good agreement with experimental data, but I wouldn't say it's a fully formed entity yet. And so please watch this space for more info around that.
Thanks, Ross. I think we'll leave it there for the questions. So thanks ever so much again, Ross, both for a wonderful talk and for your time in answering those questions. I'll just finish by advertising our next seminar, which will be given by Piers Keane on the topic of foundation models in ophthalmology. And this will be broadcast live at 1 p.m. British Summer Time on Wednesday, the 6th of September. Further details of all future talks can be found on our worldwide page, the link to which can be found in the About section of this YouTube channel. And do also subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. So thanks again to Ross. Thanks to you for listening, and see you next time on mathematical and computational ophthalmology. Goodbye.
Thank you, everyone.